Well, good morning and welcome to this live biology colloquium. This is the final in our 2019-2020 biology colloquium series here at Luther College. So this is Dr. Larson, and I want to welcome you back to campus. So today for biology colloquium, we have 4 different senior biology students, Kendra Weirens, Maggie Anderson, Maretta Hansen, and Lena Schmidt, who will each be giving short presentations about the research that they've been performing with different members of the biology department. A bit of information about how we plan to structure the colloquium today. Please keep your microphones muted if you're not already muted, unless called upon to ask a question. You probably want to select active speaker mode as well during the presentation so you can see the slides of each speaker. Following each of the student presentations, we will have a short question and answer time for that student presenter. And I'll take 2 or 3 questions before we move on to the next presenter, just so that in this interest of time, you can either raise your hand, you probably know that feature by now, or use chat to type in a question. And I think, Keister, if I'm correct, you're giving extra credit points to genetics students, right, who ask a few questions. I will mention that this presentation is being recorded today for viewing by students who are unable to join us. So once again, welcome to Bio Colloquium. And at this time, I'm going to ask Kendra Weirens to unmute herself and share her screen, and we should be able to that should be popping up here, and we'll be able to begin.
All right, so I am Kendra Weirens. I'm a senior biology major, and thank you for the introduction, Dr. Larson. I've been working with Dr. Keister for about 1.5 years now, and my research topic is looking at the requirement for extracellular matrix remodeling on one part of C. elegans locomotion known as a reversal. So the extracellular matrix is in all multicellular organisms such as humans, C. elegans. It's a 3-dimensional network of cells or a 3-dimensional network of macromolecules such as collagen, perlecan, different enzymes, and glycoproteins. It's the area in between the cells. Having the correct regulation and the mechanisms for different cellular processes is incredibly important for the extracellular matrix. And certain cellular processes such as like cell migration, cell-to-cell communication. Having the incorrect mechanisms or dysregulating the remodeling of the extracellular matrix can contribute to diseases such as cancer and neurodegeneration. Looking at some more specific proteins of the extracellular matrix. The extracellular matrix has, like I mentioned in the previous slide, different enzymes within it. Some of these enzymes are proteases, and these proteases one type of them are going to be the matrix metalloproteinase, or MMPs, and these are proteases or enzymes that degrade the extracellular matrix proteins. So looking over at this slide or this figure over here, the MMP or the matrix metalloproteinase is the Pac-Man looking guy, and it's degrading the proteins around it or the red lines. The next type of regulatory protein in the extracellular matrix is the TIMP or the tissue inhibitor of metalloproteinases, and these are proteins that inhibit the activity of matrix metalloproteinases or MMPs. And having the correct balance of MMP to TIMP is incredibly important for the regulation of the extracellular matrix. So again, looking back at this figure here, the matrix metalloproteinase is the Pac-Man guy, the TIMP is the pizza slice. If the MMP is degrading the extracellular matrix maybe too much, the TIMP will come in, kind of insert itself and inhibit that activity from occurring anymore. So it is no longer degrading the extracellular matrix around it. So my project, I was really interested in how does the extracellular matrix remodeling kind of regulate development and function of multicellular organisms? And to look into that, I looked at Caenorhabditis elegans, or a small or a microscopic nematode worm. At adult size, it's about 1 millimeter long. Like other multicellular organisms, C. elegans contains MMP and TIMP genes. Specifically, it contains 6 MMPs, ZMP1 through ZMP6, and 2 TIMP genes, Cry2 and TIMP1. TIMP1 has been shown to regulate gonadal morphogenesis. ZMP1 has been shown to be required for vulval morphogenesis, and ZMP2, if decreased expression of it occurs, it leads to decreased pathogen resistance. When some preliminary observations that I saw during my microscopy project at Luther, I was decreasing TIMPs within worms, and I noticed that they had a little bit of a locomotory deficit. They were a little bit twitchy. They just didn't seem to move correctly. So this is what interested me into going into my specific research question, which is, does dysregulating the expression of TIMPs impact locomotion in C. elegans? And in order to achieve this, I used a technique known as RNA interference, or RNAi, to decrease the expression of TIMPs within these worms. And how I did that is I took wild type N2 worms. I put them on 2 synchronous generations of TIMP1 or Cry2 feeding RNAi, which reduces the expression of Cry2 or TIMP1. And then I recorded 2 different types of videos looking at 2 aspects of worm locomotion. The first one is a 3-minute reversal video looking at worm reversals, and then I looked at, and then I wrote 30-second videos analyzing their speed, and from there on I did some data analysis. I'm going to show you guys an example of a part of a worm video that I recorded of a worm reversal. So here is the video, and the worm is going to move forward. It's going to hit that peak point of forward movement right there. Then it's going to start moving backwards, and it'll eventually curl its head upon its body, kind of forming an omega shape, and then it'll continue its now changed directional movement. So 2 types of reversals that I was interested in looking at were the short reversal, which is going to be in the top panel up here. And a short reversal is when a worm moves backwards a head's length or more. So looking at these panels, it's going to move forward, hitting that peak point of movement, which is the red dot. It's going to move backwards a head's length or more. And then it's either sometimes doing short reversals, they change direction, other times they do not. And then second type of reversal I was looking at was the omega reversal, or the omega turn reversal. And similarly to short reversal, a worm's going to move forward, hit that peak point of movement. It's going to go backwards, and then it's going to start curling its body upon itself, making an omega-like shape, as pictured with these little omega shapes within the second panels here. And then it's going to change direction most times and continue on its way. The next way I looked at worm locomotion was looking at path lengths, and these were generated using the ImageJ plugin WormTrack. So it takes videos of worms and it just kind of plots out their movement throughout a period of time. So I looked at the control worms, the Cry2 and the TIMP1 treated worms. And looking at their worm paths, a red asterisk indicates a reversal occurring within the path. So just looking at comparatively between the paths that are chosen here, you can see that worms treated with Cry2 and TIMP1 RNAi do exhibit a higher level of reversals during the time periods shown in the bottom left corner here. So my data, I looked at the worm reversal frequency, as I said before, and it shows here that worms treated with the Cry2 RNAi exhibited a significantly higher reversal frequency when compared to the control worms. And while TIMP1 had a similar trend to Cry2, there was no significant difference between the control worms. To analyze worm locomotion more, I wanted to see if the RNAi actually, or the reduction of Cry2 and these TIMPs had a significant impact on overall worm locomotion, if they were still affected, if they could get around fine, if their overall sinusoidal type movement was affected. And in order to do that, I looked at average speed. And the average speed, I did not see any difference between the two treatments and the control. And so that just means that with that, they can still move around fine. They just have an increased reversal frequency during that time. So with this phenotype of increased reversal frequency caused by a decreased expression of cry2, I was curious on how does decreasing cry2 expression overall affect worm locomotion. And to do that, I was looking at where it was expressed within the worm. So to do that, I used some cry2 transcriptional reporter strains. And a cry2 transcription reporter strain works by regulating the expression of a fluorescent protein using cry2 promoter. So here, the cry2 promoter is driving the expression of either GFP or M-neon green in my worm strain's case. So I used two different worm strains. I used BC13623, which is the cry2 transcription reporter regulating the expression of GFP, and then I used HIE201, which is a cry2 transcriptional reporter regulating the expression of M-neon green. I used these two strains because HIE201 was a little bit brighter, and it allowed me to confirm and compare the two strains for where cry2 is expressed within the worm. So here is a picture of overall cry2 expression within BC13623. The anterior side of the worm over here is going to be where its head is, and the posterior is going to be where its tail. The dotted line indicates the outside of the worm. And the methods that I used for my microscopy was I took these two worm strains, BC13623 or HIE201. I paralyzed them using a paralytic known as levamisole. And then I put them on the wide field fluorescent microscope. I took some images, and then I manually looked at them using the image, and then using ImageJ. So where I saw expression was within widespread was within body wall muscle and muscle cells in general. So here's the first one, body wall muscle. And here's a picture of it in the BC13623 worm strain. The brighter spot here is going to be the nucleus of the cell, and then these little projections here are the muscle arms. And the outline, the dotted line here again, is indicating the outside of the worm. The second type of muscle that I saw expression in was vulval muscle. So here's a picture of some vulval muscle cell within the worm. The dotted line here indicates the outside of the worm. The arrows are pointing towards the vulval muscle. The small triangle here is pointing towards the vulval slit, and the dotted lines are, again, the outside of the worm. The next area that I saw expression in was some cell pairings within the head and tail. So here they are in the tail. These arrows here indicating the cell bodies, and in the HIE201 strain, we did see some projections coming off of the cells here that are indicated by the small triangle, and the dotted line again is showing the outside of the worm. And we also saw some cell pairings within the head. So outside here again is indicated by the dotted line. Some cellular projections are indicated by these triangles. The two asterisks are showing the anterior and the posterior bulb of the pharynx in the worm. And we believe that these cell pairings that we are seeing within the head and tail to be sensory neurons due to having a similar morphology as sensory neurons. So the broad conclusions of my research are decreasing expression of cry2 increases reversal frequency. Cry2 is expressed within muscle cells and sensory neurons in the head and tail. And a future guiding question is the regulation of the extracellular matrix by cry2 important for the development of sensory neurons in C. elegans? And going into more with that future guiding question, I have a few future directions in order to pursue after this project. One is to confirm an RNAi phenotype with genetic deletion strains. Some initial studies were done, but the results were variable and inconclusive. So continuing into that, we would be able to confirm our phenotype that we saw with our RNAi. second one would be to confirm that the cell pairs that we are seeing within the head and the tail are sensory neurons. And so what we could do for that is we could mate a species of a sensory neuron fluorescent strain with a Cry2 transcriptional reporter and compare that the areas that they fluoresce in are the same. And the third would be to investigate the impact of reduced Cry2 expression in sensory neurons. So this would be using GCAMP, which is a calcium indicator, to see if neuronal function in worms is normal with a decreased Cry2 expression. So I'd just like to make some acknowledgements quickly. I would like to thank Robert Fitton for all help with their microscopy. I would like to thank all the other Heister lab students, past, current, Rye Holly, Rossier, Gabby Blair, Graham Hoshaw, Nina Kubmova, Isaac Higgum, Nor Beatty, and Greg Martins. I would like to thank the Luther College Dean's Office for providing funding over the summer for my summer research project. I would like to thank my Bio 490 class for all of the help they had with my presentation and with my paper. And then I'd like to thank the Luther College Biology Department for providing all the materials and as well as funding for my research. Does anyone have any questions?
So Hamad, you can either unmute yourself or type it in the chat. There. Somebody typing out a question. Any hands up? Anybody want to ask a question there? So there's one on a 6:2 ratio between the TIMP and the MMPs. Did you see that, Kendra?
Yeah, I see the question there. So I'm not sure why there is a 6:2 ratio between the TIMPs and MMPs. The TIMPs just have, I guess, a wider spread for inhibiting the different MMPs there. And I'm not sure if it is the same for all organisms. I'm going to make an assumption here that it's probably not due to different organisms just having different regulatory methods and a different kind of complexity between each of them.
Dr. Enos.
Yeah, that was a great talk. Thank you for that. I had a question about two kind of technical questions. I'm wondering if you could explain a little bit more about how the feeding of the RNAi procedure, how that works, and then also the reporter strains with the GFP or the other one. Are those, I'm curious about copy number. Like, is that just one copy that is in addition to the wild type that's on the chromosome? So yeah, just.
Okay. So RNAi, feeding RNA, the very basic explanation of it is, so we have bacteria on these places these worms like to eat, and we take bacteria and we have like a plasmid in it with this certain gene within it that, and they eat it and it knocks it down. It's a little bit of a complex mechanism that even I'm not fully clear on the exact molecular basis of every single step within it. With the transcriptional reporter strain, one of them was actually one of them that Dr. Heister made himself. And I'm not fully sure how he made it actually, but I know that they it's a similar, I believe, method where they're just edited and yeah, I'm sorry, this isn't the greatest answer.
That is okay. I'm asking pretty technical questions, and these are things that are probably embedded before your project even started, but they're just out of curiosity based on me living in a different world that the bacterial world and thinking about how we're doing things in there. Thank you.
Yeah, of course.
So Kendra,
And I see, oh, Robert has a question today. Answer that real quick or should we move on?
great. And then
Okay.
we'll go on to the next one, but answer that one.
Okay. Robert Fitton asked what kind of troubles I had with video analysis. So figuring out the ImageJ plugin worm track was a little bit of an issue because that was something that no one at Luther had really any current experience in. So I had to do some digging into figuring out how to use ImageJ and the ImageJ plugin, which luckily I was able to figure out. And then I also had some problems in the beginning with getting the videos that I recorded off of the microscope because they were in a different format than ImageJ liked to use. They were in MP4 format, and ImageJ likes to use AVI and TIFF formattings. So I had to get a plugin called FFmpeg to work on ImageJ in order to convert these videos before I was able to really do any analysis.
All right. Well, thank you,
Easier
Kendra.
said than done.
Yeah. All right. Well, thank you, Kendra, very much. If you want to stop your sharing there, and we'll ask Maggie to go ahead and pull her slide up. And we got Maggie Anderson doing our next presentation here. Let's see.
Can you see it?
Yeah. Yep, we can see it.
Okay, cool. Okay, hi everyone. My name's Maggie. I'm a senior biology major, and I spent last summer working with Dr. Larson studying the ground beetle communities of Anderson Prairie and Lionberger Environmental Preserve. This is a ground beetle. It is an insect, so like all insects, it has a head, a thorax, and an abdomen, 6 legs, 2 antennae, and 2 pairs of wings. It's in the order Coleoptera, so that's the beetle order, and that's characterized by hardened front legs called the elytra. And then it's in the family Carabidae, which is characterized by the segmented antennae and 5-5-5 tarsal segments, which is the hind legs. Other families of beetles will have different types of antennae and tarsal segments. Ground beetles are a really large, abundant, and diverse family. So with 20,000 species worldwide, many are predators, so they eat other insects, which is crucial in many food webs, and also as pest control, especially for organic farmers. Some species of ground beetles are habitat specialists, so depending on the species presence and their abundance, it's possible to determine ecological and environmental quality. Ground beetles have also been studied in Northeast Iowa for the past 20 years by Dr. Larson, and about 200 species have been identified, so there's a really great baseline knowledge of ground beetles. In addition, insect declines are becoming more and more well documented. It's often termed the insect apocalypse in the news and even journal articles. You may have heard of monarch butterfly declines and also honeybee colony collapses, but ground beetles have also had documented declines. This study came out in 2012. It was performed in the United Kingdom, and it found that 75% of the ground beetle species there had declined over a 15-year span. So the objective of our study was to document the composition of and compare ground beetle communities in Decorah, Iowa at 2 locations, Anderson Prairie and Lionberger Environmental Preserve. So here's our 2 sites, Anderson Prairie on the left. We had plant habitat, we sampled from plant habitats here of planted prairies with different burn regimes and mowing as disturbance. And then Lionberger Environmental Preserve, we sampled different habitats of forested, grassland, and riparian. Here's a map of Lionberger in relation to the Luther College campus. You can see some right there, and across Highway 52 and follow Pole Line Road, you'll get to Lionberger. It's really cool. If you haven't been there, I'd recommend it. To collect our ground beetles, we use pitfall traps. In the upper right-hand corner, you can see it's just a plastic cup, and we place it in the ground so that the lip of the cup is even with the surface of the ground. You can see it placed here. And then we fit the plastic cup with a funnel to prevent beetle escape and fill it about a quarter full with propylene glycol as a preservative. Dr. Larson and I made these covers to place over the trap and about an inch off the ground to protect the trap from rain and disturbance from predators, mainly raccoons. And then we would always put a little flag next to the trap so we could easily recover them. The pitfall traps were set up in transects, and one transect is 4 pitfall traps set about 10 meters apart. So we place a trap, walk 10 meters, set another one, and so forth until we get 4 in a straight line. The 4th one is off the screen in this photo, but that's what in each plant habitat. So this is our Anderson Prairie site, and our plant habitats we sampled here was prairie burned in 2019, prairie unburned since at least 2017, and prairie mowed in 2019. So we had a transect and prairie burned in 2019 here, here, and here, and the same thing for all the other plant habitats. Here's Lionberger. We have our floodplain forest, basswood, plant habitats, oak hickory forest, old pasture prairie, and reed canary grass, each with 3 replicate pitfall trap transects. We leave the transect field for 40 and then go and pick them out. And in the right-hand corner, that's kind of what a trap would look like when we get it from the field. You can see ground beetles on top, but then there's also other things in the trap as well. Just whatever happens to be walking across the ground and falling into the trap, that becomes part of our sample. So I would carry all the traps back to the lab in this crate in the background and clean them out, get all the dirt out. And then once the samples were cleaned, we stored them in ethanol for a little bit. Then I would dump the samples out underneath the microscope, pick out all the ground beetles, and we would pin them up like this, identify them to species, and count them. So overall, we collected a total of 1,194 beetles representing 60 species. This photo in the background is our more official collection, and you can see the diversity in ground beetle size. Each little grouping is a separate species, so there's some large, some small, relative to each other. Trasticus stygicus was the most abundant species collected, and it was a forest specialist found only in Lionberger forest habitats. And Agonum palustre was a wetland specialist. All but 1 specimen were found in the reed canary grass and floodplain forest, which are both really wet areas. It's kind of hard to tell in this photo, but Agonum palustre is a really tiny beetle, while Trasticus stygicus is a little bit larger. Cyclotracheus sodalis and Poecilis leucoblandus are both generalists found in every single plant habitat we sampled at both Anderson Prairie and Lionberger Environmental Preserve. On the left is Clanius platyderus. It's a grassland specialist found in all the prairies and grasslands at Lionberger and Anderson, while Trasticus mutus is a forest specialist found in none of the prairies at Lionberger, Anderson, but in all the forests at Lionberger. So here are results. Overall, we found a higher beetle abundance, standardizes beetles per trap per day at Lionberger Environmental Preserve on the left, and lower in Anderson Prairie. When we did the statistics, our NOVA and post hoc tests, we found that the reed canary grass and maple basswood forest at Lionberger were the only 2 that were significantly higher. Same thing for species richness. Overall, Lionberger Environmental Preserve had much higher species richness compared to Anderson Prairie, except for maple basswood forest. Once again, ANOVA and post hoc, we found that 4 out of the 5 at Lionberger were significantly higher than Anderson Prairie. And it's interesting to note that the reed canary grass at Lionberger had high species, beetle abundance and high species richness, while maple basswood forest had a high beetle abundance but a low species richness. That's because the maple basswood forest was dominated by 2 species. This is a principal components analysis, which takes the number of species and their abundance and graphically positions our plant habitats based on how similar they are. So the points that are closer together are more similar to each other, while the greater distance between points on the graph are less similar. So our forest habitats right here have similar ground beetle communities, and our grasslands also have similar ground beetle communities. The reed canary grass is definitely an outlier, really far away from all the other habitats we sampled, and that's because it was dominated by the ground beetle species Agonum palustre. So overall, this research provides a really good baseline for Lionberger in 2019. That's the first time any beetle sampling has been done there. And the data that we collected for Anderson Prairie can be added to Dr. Larson's long-term studies of ground beetles in this area. When we compared Anderson Prairie a little bit to what Dr. Larson's collected in the past, we are noticing trends in decline in number of species and their abundances. So in the future, as climate change continues, it's going to be really important to continue monitoring ground beetles, especially in this area. I would like to thank the Luther College Dean's Office for their support funding this project, and to Dr. Larson for his guidance and help all throughout the summer and school year. I really appreciate it. And thank you for listening, and I'd be happy to take any questions.
Once again, you can put up your hand or type in a chat question. Thanks. Any questions for Maggie?
Did one
Sorry.
So, go ahead, Maggie. Read the one question there.
I can't see it.
Okay. So Haley's asking you, how did you do the transects exactly?
Oh, exactly. Okay, yeah, so we have a little digger. It's in the shape of a plastic solo cup, so it's narrow at the bottom, and we just stomp that into the ground, take out a little core of dirt, and then just place the cup into the ground with the funnel in it. And then we just leave the core of dirt sitting there for the 2 days that the trap is out there. Take the trap out of the ground, bring all the materials back, and just place the core back into the ground. So it's like the trap wasn't really there at all. So when we place one, walk 10 meters in a straight line, do the same thing for the next, and so forth until we get 4 traps in the ground.
So we've got a couple others. Brooke Stanga's asking, do you think that these results would be similar to the rest of northern Iowa?
Yeah, that's a good question. So Dr. Larson's research in the past has covered a wider area. He found that the forested and prairies that he collected beetles from compared to ours today are higher. So I think overall, yeah, I think that's safe to say, but it's always kind of hard to generalize everything for a really broad area. Are the ground beetles indigenous to the area, or were they introduced? Yeah, many of them are just native here. Lots of specialist species in both Lionberger and Anderson Prairie. So that's indicating that, yeah, we've got good habitats here. They're supporting many ground beetle specialists, but yet there are still fewer specialists than there were in the past, we're noticing. So possibly habitat declines. What are some of the challenges you faced during your research? I guess the hardest part was identifying the Bembidion quadrimaculatum. That is a really, really tiny beetle, and it looks really similar to another species. So we actually had to start doing some genetic testing on it to determine the difference between the 2 species. So I think that was the most challenging part, figuring that out like halfway through the summer.
All right. So one last question here from Noor. The presence of similar species of beetles depend on the type of soil or type of plants?
Yeah, that's a really good question. So our principal components analysis showed that the forested habitats were providing for different types of beetles compared to the prairie habitats. So yeah, the type of plants does matter. I think we, in the future, should probably go out and do surveys of the plant habitats a little bit more to determine the type of species that are present there, to really say like what types are affecting them. But we do know that there are differences.
All right. Well,
Thank
thank you. Thank you, Maggie. We're going to move on to Moretta. So Moretta, you can go ahead and unmute yourself and great.
All right. So my senior research project focused on PI3K AKT signaling and its influence on dopamine function in C. elegans. And so today I'll be giving you a presentation of a poster that I actually presented last week at a national genetics conference. So starting from the beginning, PI3K AKT signaling is a subset of the entire insulin signaling pathway. So as you can see in this little diagram here, this is really what we're focusing on. When a ligand binds to the receptor here, it causes a conversion from PIP2 to PIP3, and then a different process converts it back from PIP3 to PIP2. But downstream of this signaling, so when this is active, there's at least hundreds of factors downstream that it impacts. And one that we're really interested in that we don't know much about is dopamine. So PI3K AKT signaling has been observed a lot with regards to cancer because being part of the insulin signaling pathway, it's pretty critical for cell growth. And if it goes unchecked or out of control, it can cause cancerous cell growth where the cells just don't die when they're supposed to and continue growing and invading other tissue. But we also notice that when this is unchecked, there seems to be something happening to the dopamine function. So using C. elegans, which is the same model organism that Kendra used in her project, we performed a couple of different experiments to try to determine how or if this excess PI3K AKT signaling or even a lack of this PI3K AKT signaling actually influenced dopamine function in these animals. So the first experiment that we did, this is called tap habituation. And similar to Kendra's experiment, it measured the reversals of these animals, but this time what we were measuring isn't whether they reverse at all, it's whether or not they reverse in response to a tap stimulus. So if you think about yourself, if someone taps you on the shoulder with a controlled amount of force over a specific amount of time, at first you're going to respond pretty strongly because it's a new stimulus, but as it continues with that recognizable amount of force over a recognizable amount of time, you stop responding. So this is your body habituating to that tap. And so that's what we're measuring in these animals. And so the results of this first experiment can be seen in the middle bottom of this poster here. And what was interesting is that this purple here, this is our wild type, our normal animals. And this blue, these are animals that are dopamine deficient. So we see that these blue animals, they don't respond similarly to the wild type animals, which is kind of what we'd expect because tap habituation is a measure of dopamine function. So dopamine is a neurotransmitter that's critical in both movement and learning. So learning to stop moving depends on dopamine. But then if we look down here, these are our animals with different PI3K AKT signaling. So in orange, these animals have less PI3K AKT signaling, and they actually respond pretty similarly to the wild type, the normal animals, and they actually respond a little better, which is interesting. But these animals with excess PI3K AKT signaling, so they're having too much signaling, they respond a lot more similarly to these animals with dopamine dysfunction rather than to the wild type or normal animals. So we're seeing some sort of link between an excess of this signaling and problems in dopamine function. So our second experiment just confirmed this. This is glyoxylic acid induced fluorescence. So basically by putting these worms under certain conditions using glyoxylic acid, we're able to cause their dopaminergic neurons to fluoresce. So this occurs through glyoxylic acid interacting with dopamine in these animals, and it locates at dopaminergic neurons because they contain so much dopamine. So looking up here in the top middle, we see that our wild type, our normal animals, they do have dopamine neurons that fluoresce under these conditions, which is expected. These animals here in orange, they also have dopamine neurons that fluoresce, which is also expected because, again, these animals are ones that have less insulin signaling activity. What's interesting is over here, we'll go to the far right, these CAT2 animals, these are ones with impaired dopamine function. We don't see dopamine fluorescence in these animals, which we could kind of expect if they have impaired dopamine function. But these DAF18, these animals with excess insulin signaling, they again look like the animals with impaired dopamine function, which is interesting, and it confirms what we're seeing down here, where somehow this DAF18, these animals with excess PI3K AKT signaling, have are more similar to these animals with impaired dopamine function than to the wild type or normal animals. So our final experiment was HPLC, high performance liquid chromatography. And so this is a way to quantify the amount of dopamine in these animals. And so that can be seen over here. So our wild type, our normal animals, they establish a baseline here of the amount of dopamine we would expect. And the as we can see, the DAF18 in age 1, these ones with impaired insulin signaling do have less dopamine, but unfortunately with everything going on, we weren't able to collect enough samples to determine if there was any statistical significance from this. So we can just guess that there is something going on here, that insulin signaling impacts the amount of dopamine in these animals. So from all of this, we can conclude that in some way, these DAF18 animals, these ones with excess insulin signaling, have an impaired dopamine function because they look a lot more similar to these CAT2 animals. And in the future, what we'd like to look at is how does this DAF18 or this excess insulin signaling actually impair dopamine? Does it stop the synthesis process? Does it impair dopamine receptors? Does it do something else that we haven't really thought of yet? So I'd like to thank the McElroy Foundation for their research grant, Luther College for research funding, and everyone else who's been a part of this project, especially Dr. Fredham for her guidance. And thank you for listening, and let me know if you have any questions. Thank you.
So yeah, feel free to unmute yourself and ask a question, or type in a question in the chat function as well. Moretta can answer those.
Hi, Moretta. I have a question.
Yeah.
So has anyone looked into whether or not any components of the dopamine synthesis pathway, if any of those components expression is regulated in some way by insulin signaling?
Yeah, so not that I know of. The insulin signaling pathway hasn't really been looked at yet with regards to dopamine specifically, just because all of its focus is on cancer. Mm-hmm. But that's definitely something that would be interesting to look at, because this pathway is involved in regulation of all cells, including neurons, and especially dopaminergic neurons.
Yep.
Any other questions for Moretta? Let's see. There's one there in the chat. Yeah.
Yeah, so this was honestly my favorite part, or one of my favorite parts of the project. So using some funding, I was able to buy a Lego robot that I then built to tap this plate. So I was able to program this robot to tap the plate once every 10 seconds, and it tapped with a controlled amount of force over a controlled amount of time. And so we were able to record this just using a standard video camera, and then run everything through a worm lab, which is a computer program that actually quantifies reversals and things like that.
Hi, Mariota. I have a question too. So I was wondering, you used the HPLC test to quantify the amount of dopamine?
Yes.
In the graph, it's lower than, the DAF18 is lower than the wild type. And I thought that kind had more dopamine. Can you kind of explain why that was the result?
Yeah, so overall, we're actually expecting that these DAF18 animals would have less dopamine than wild type. Because again, these are the animals that we would expect to have impaired dopamine function in some way. I think The age 1 is an interesting result because these are the animals that actually seem to have better dopamine function in our tap habituation, and they actually respond more similarly to wild type. But this, unfortunately, like the N here was only 4 to 5 samples, so we really can't draw a whole lot. This is one of our more preliminary results from this, but it does seem to look kind of like what we'd expect.
Okay, thank you. Sorry, I meant to say age 1. That was my mistake.
Yeah.
But thank you.
Yep.
There's one other question from Charmaine, and then we'll move on to Lena.
Oh yeah. So the technique. So the tap habituation technique actually did save a significant amount of time. So I was able to actually program the robot and let it run for about 10 minutes. The worms had to just sit there and acclimate to the plate. And then while it was doing the tapping for 5 minutes, so that's 15 total minutes, I could be doing something else, which was really great because I ran about 75 plates, and 75 times 15 is a lot of minutes. But then also running everything through Worm Lab saved a lot of time because I didn't have to watch each worm to determine if it reversed, because this program would do it for me. So yeah, definitely technology is pretty great for saving time in research.
All right. Well, thank you, Moretta. Nice job. Let's, Lena, you can start sharing your screen and unmute yourself. And
Get this into slideshow. It'll work for me.
it's
Come
coming.
on.
It's working.
It's getting there. There we go. All right. Hey, everybody. I'm Lena. I'm a senior biology major, environmental studies minor. And I have been working for Dr. Larson in the entomology lab for the last 2-ish years. And we have been studying moths, specifically of the oak hickory forests and planted tallgrass prairies on campus. So the main objective of this study was to compare the abundance, species richness, and composition of moth communities in planted tallgrass prairies and oak hickory forests on the Luther College campus. But we also just wanted to find baseline data because nobody has studied moths in the past here in this area, or at the very least no Luther projects have studied it. There was one guy in like 1908 that did a study, but that's not super helpful. So we wanted to get that baseline, what do we even have for moths, so that long-term studies can be done in the future. So we sampled in 6 different locations, 3 planted tallgrass prairies, so Anderson, Gateway, and Jewel were our prairies, and then 3 oak hickory forests, so Hickory Ridge Woods, Spildy Woods, and then again Lionberger Environmental Preserve, way out here. And the way we did this, unlike Maggie, we didn't use the pitfall traps because moths can fly. So we used blacklight bucket traps instead. And these do exactly what they sound like. There's a blacklight in the middle. The moth is attracted towards that, flies at it, hits a plastic screen, and falls into the bucket where we can take it back to the lab. But like the pitfall traps, these can also collect a variety of other things besides moths. So in the top left corner, you can see what a average bucket would look like after we brought it back to the lab. So you can see there are beetles in there along with the moths. We also got a ton of caddisflies, a couple traps, and some wasps. So my next job was sorting all of that out and sorting the moths into species based on appearance. So moths that look the same, we assumed were in the same species. Next, a good portion of my time was spent pinning and spreading moths. So this was done not only to make it easier to identify them, but also make them more displayable. And I did this basically for the entire summer of my research. And then after that, we had to identify and label all of those moths. So a huge shout out here to Abby Miller. She's in the top picture on the right, helping me identify, and also Jim Durbin on the bottom, who was a huge help identifying all of the really hard to identify brown moths. All right, so what we found, we had collected a total of 12,290 moths, which is a lot. We managed to identify about 80% of those, so 9,560, and these represented 34 families and 468 different species. So as you can see, we had a couple of unique families and species in the prairies and the woods. So there were 4 unique families in the prairies, 15 unique families in the woods, and then 77 species that were only found in prairies and 248 species that were only found in the woods. And then we also managed to find quite a few state and county records. So for those of you who are unfamiliar, a state record would be a moth that hasn't been found in the state of Iowa before. A county record would be something that hasn't been found in Winneshiek County before. And we managed to find 7 state records and 97 county records, which was awesome. So here is a comparison of the prairies and the forests. On the left, you're going to see number of moths. On the right, you're going to see species richness, so number of species of moths. And as you can tell pretty clearly, there were a lot more moths and a lot more species of moths in the forest than the prairies. Going back. Okay, in the prairies, we had an average of 872 moths. In the woods, there was an average of 3,224 moths. So about 4 times more moths in the forest than the prairies. There was also in the prairies an average of 129 different species of moths and an average of 251 species of moths in the woods. So about 2 times more in the forests. More species in the forests. So these graphs are showing similar things. We've just split it out into each individual site. And again, as you can see pretty clearly, there are a lot more species of moths and moth abundance in the forests. But what you can also see here is that within the habitat types, within the prairies and within the woods, the sites are very similar. So for example, all of the prairies have very similar numbers of moths. All of the prairies have very similar numbers of species of moths, and the same for the woods. And then, like Maggie, we did a principal components analysis. So again, the way to read this is basically the closer together the points are, the more similar those sites are in species, like the types of species that are there and the abundance. So as you can see pretty clearly, the prairies are grouped very closely together. That means that they are very similar. They have very similar moth compositions. And then over here, you can see the forests are a little bit wider spread apart. So they had a little bit more variety in the species and abundance of species that were there. But you can also see that the prairies and the forests are in 2 very distinct, separate parts of the graph. So that by that we can see that the prairies and the woods had very different, distinct compositions of moths. So some of the most common moths that we found in the prairie were the most common was the pale lichen moth with 145 individuals, then followed by the bristly cutworm, the large ruby tiger moth, the confused dagger moth, and the harnessed tiger moth. Then you can compare that to in the forest, the most common species. The most common was the hickory leaf roller with 401 individuals, followed by the pink-barred pseudostrotia, the horned spanworm, the gray archips, and the zigzag herpetogramma. So you can see even here, the 5th most common species in the woods, the zigzag herpetogramma, had more individuals than the most common species in the prairie. So just another representation of just how many more moths there were in the woods. And then just for fun, some of my favorite moth species that we found were the spotted apateloids, the grape leaf skeletonizer, Harris's 3-spot, the rose hooktip, and the Luna moth. So those were all really fun to see. So in conclusion, we had 4 times more moths in the woods than the prairies and 2 times more species of moths in the woods than the prairies. And we're starting to think that this might have something to do with the diversity of the plant life at those locations, but a little bit more research is going to have to go into that before we can say that for sure. And then the moths in the planted prairies were quite different from the moths in the forests.
and you can always find a ton of moths and new species records in areas when you look for them. So I would like to acknowledge again Abby Miller for helping to organize, identify, and database our entire moth collection. She's a current sophomore in the entomology lab. She's doing fabulously. And then also Jim Durbin, especially for his help with identification, but also for his Insects of Iowa website, which was pretty crucial to the identification process. Dr. Larson, of course, for his help for the last 2 years and watching me get frustrated over very small moths. And then also we'd like to thank the Luther College Dean's Office for providing funding for the Summer Student Faculty Collaborative Research Project. So with that, does anyone have any questions?
So there's a question in the chat there, Lena. I don't know if you can read that, about
All right,
what did you do with all the other insects besides the moths?
Unfortunately, well, a couple of them got moved into my collection, but most of them ended up going in the trash, which is unfortunate. But there were so many caddisflies. There were just so many, and they get squishy much faster than the moths.
Elena, I have a question.
Yeah.
Your trap, well, a couple things. Number one, I just really enjoyed hearing the names of all the moths, all the sort of informal names. They're fantastic.
There are some really good
I think
names.
the zigzag one was particularly fun. I was curious about your trap.
Okay.
Do you think that it actually captures all moths equally, or do you think that maybe some moths are trapped more so than others, and to what extent do you think that might affect the representation of moth species within your different areas?
Yeah, that is a great question. Blacklight bucket traps are a pretty standard moth trapping equipment, but there have definitely been some studies done that have shown that some moths are attracted more frequently than others. Specifically, the family Noctuidae, I believe, is one of the ones that's overrepresented. There are also quite a few moths that fly during the day, and so we're not picking up any of those. But overall, since it's such a standardized way of doing things, it's at least comparable to most other studies.
Okay. How would you trap the moths that fly around during the day?
That's a good question. You can use pheromone traps for some things, but that's more of a specific, it's often used as a pest control. Yeah. Some places will use like sugar trapping, so you can like spread like a sugar mixture on trees, or I'm not super clear on exactly how that works, but there are definitely other ways to do it.
Cool. Thanks.
Yeah. All else fails, you go out there with a net.
There's a question here from Michaela.
Okay. Is there anything other than plant differences between the prairies and the forest that you think led to the huge difference in number of moths? Yeah, so the plant difference is going to be the main one, because moths, like beetles, are often specialists, so the larvae, the caterpillars, will oftentimes eat only one type of plant in their life. But other things that have been found to affect moth differences are moonlight. We did try to minimize the effect of moonlight by trapping on specific days when the moon was less full, but also just the enclosedness of the forest would make the light travel less far. So it's kind of conflicting. We're guessing that the main reason is the plant types. Mm-hmm. Did I answer your question? I'm sorry, that was very back and forth.
Yeah, you're good. You're good. I was just curious, just like from a interest standpoint. I just thought it was interesting that there is such a huge difference in numbers. So I knew you didn't know for sure, probably like you said at the end of your presentation, but I was just interested in it. Thank you.
Yeah. Okay, what else do we have? Does the species richness and diversity compare to any other research done in Iowa, Minnesota, Wisconsin? That I don't know. This is the only real study that I know of that's been done in this area.
Yeah, there's not a lot of studies like yours that have been done. So
Okay, one more. Since you did such an extensive research with moths, what's the most exciting thing you learned about moths besides the diversity of colors and beauty? I do like the fact, like some of the, especially the big Saturniids, this is more of a fun fact than anything else, but some of the big Saturniids, like the Luna moths, don't have mouths as an adult. So they spend their entire time as a caterpillar just eating and eating and eating and eating. And then as soon as they pupate and turn into adult, their sole purpose in life is to mate and then die. And they usually don't have very long to complete that because they can't eat, so they only have so long to survive. So I thought that was really cool. My favorite is the inconsolable underwing. Thanks for coming, you guys.
Well, thank you, Lena.
Yeah.
So this will conclude our biology colloquium today.